Okay, so first of all, thanks a lot to the organizers to gather to putting together this kind of event. It was about 6 months ago when our hearts just frozen. Somehow we got the news of Pepe's passing away, and that was very upsetting. During these 6 months, many things have happened, many things have been written, and every time two persons that came together and that knew Pepe just met, I don't know, he was the subject of our conversations, and we were all the time just remembering him. So thanks the organizer for inviting me as well. So it's a great pleasure to be here with all Pepe's, most Pepe's friends and collaborators, and this is a pleasure, and it's really touching having this opportunity to talk now. Many things have been written about him. I've allowed myself to make some kind of tag cloud of based on these nice obituaries, and I think that looking at this cloud we can have, I don't know, at least I see a little bit of Pepe's profile. So that was what I have chosen. Many pictures have been shown, have been shared this time, and I've chosen these 4 pictures because to me they just picture very well how was Pepe as a mentor, as a PhD thesis director. The great atmosphere in the lab, this basically, great time in the lab. So we had the opportunity in these early stages in Pepe's group to explore almost everything. Everything was allowed. Every problem can be addressed, can be studied, and we were all together working in such different things like nitrogen metabolism, transposition, genome organization, methylation, gene regulation. I don't know. I myself started working in nitrogen regulation, then moved to transposition, then moved to recombination, then whatever. So that was a real happy and very rich time for research, for our growing as scientists, and we will keep that those learnings during all our lives with ourselves. The good atmosphere in the lab was not only in the lab, just moved in over beers and food and just discussing, and so that was a great atmosphere. He also took care that we get involved as students, as PhD students, with all the invited scientists he brought to Seville. He was very active, bringing really top scientists, and we were always involved in just discussing with them, going for beers, and that's something that has been part of our learning as scientists during his the time we could share with him in our thesis, in our PhD thesis. He also helped us as a mentor to many other things. In my case, to choose my postdoctoral stay. So he helped me to find a very fine laboratory. I went to the University of Utah to work with Sandy Parkinson, and here's Pepe. Visiting us in my apartment there in Utah, and a very special day. My first daughter was about to be born right there. She's from Salt Lake City, and that has always been very helpful, and has been always taking care of us even years after we left the lab. When I came back to Spain from Utah, I went to the University of Malaga. I joined the group of Juan Jimenez, which is here in the audience, and I moved working with GIST. So I kept a little bit, not as close to Pepe scientifically as I had been till that moment. But we always had the opportunity to gather for lunch and to discuss science at least every year or every other year. And I have to thank Amanda Flores, who is here, that made great efforts for us to keep in touch together. And now I'm supposed to talk about some science here. Of course, I'd love to. And since I came to Spain, I've been working in GIST, but as the audience here, the theme is bacterial genetics. I have to say that I never stopped doing some bacterial genetics. Somehow, even working on GIST, I've always had found the opportunity to collaborate, to make collaborations with people, just trying to apply what I really have learned with Pepe and in my first years in bacterial genetics. So, what to tell today? What story I want to tell today? And now here's where my talk starts. I just going to refer to the last time I met Pepe. I was talking to him. I had the opportunity to have lunch and to discuss science over beers, and that was May 2021. That was the last time I could talk to him. I had the opportunity. Amanda was with us too. And as you may imagine, everything we talk about, many things about politics, about science, about life, about wine. And so to choose something to talk today about, and I don't have many time, but that's fine. So I'm not going to talk about politics or economics. That's not my field. I'm not going to talk about wine. Unfortunately, this is more a GIST guy thing. So sorry, folks, because this is bacterial genetics. So I'm going to talk about collaboration. I'm now asking with a group asking bioinformaticians asking about the real biological significance of the CRISPR system in bacteria. So I've called these sketches drawn from my last scientific conversation with Pepe. And in fact, this is the two smart guys. I'm really very smart guys. I'm really lucky to collaborate with them, Antonio Perez and Alex and Rubio. So these are the real smart guys, the ones that are holding the projects. I'm the one having fun, just discussing with them and collaborating. So what's the question? The question is about the biological significance of the CRISPR-Cas system in bacteria. So we work with all the genomes from this ESCAPE group of bacteria. Many genomes have been sequenced so far, and this is a wealth of information that we can use to ask any kind of biological question. So first of all, is CRISPR widespread? Is such a nice invention for bacteria that every bacteria needs it. In fact, this is no big surprise, but we find that it's not really very widespread. So CRISPR systems are not really everywhere. Okay? So only a subgroup of bacteria in any of the groups have that. Okay. So what's that for? Okay, that this is no big surprise. Okay, so if we search the spacers, the targets that the CRISPR system has in the to fight, so we find that basically phage and plasmids are the most abundant spacers that we can find. One thing that we can see here is that if we do this pangenomic analysis, if we check all the genome sequence, what is called the dark matter is really reduced. So it's published normally that 80% of the CRISPR spaces is dark matter, not targets identified. But in fact, if we analyze all the pangenomes or all the genomes published, we can reduce this. No surprise. Basically, most of the spacers are against phage or these called phage plasmids. CRISPR are not really widespread. So is that recent acquisition? It's a phylogenetic branch of the bacteria that has this system, or it's widespread. And doing phylogeny with core genes, this is MLST, okay, of all the genomes that we have analyzed, we can see this kind of hide and seek or peekaboo game of the CRISPR-Cas system. So it's been acquired and lost many times, right, during the phylogeny. So it's not that I gain the CRISPR system and I keep it because it's very good. In fact, I acquired that. It's useful for something, but when it's not useful, I better lose it. Okay, some kind of things. This is with the core genome, but if we do the same analysis with the gene profile, which takes into account all the pangenome, all the genes in the pangenome, presence of absence, we have the same picture. Okay, so in fact, and this is where in the point when I have discussing this with Pepe, and somehow it looks like the acquisition of CRISPR-Cas system was like an indirect trade, giving some indirect advantage. So when for something, when that something is not necessary, then the CRISPR system is lost. Okay, so what is this something? So let's check for genes that come along with CRISPR-Cas system. So the strains, the genomes that harbor this CRISPR-Cas system. So what kind of genes have more often? Right, and well, I'm showing here just for time only the example of Acinetobacter baumannii, but we have done that in all the genomes of the ESCAPE system, and here we go. So basically, the CRISPR-Cas system comes along with some membrane proteins. Okay, most frequently. So now we have a relationship here. So we have CRISPR-Cas system with membrane proteins. Some ideas start building up. So what has to do membrane proteins with CRISPR-Cas that is mostly directed against phage, right? So is it possible that CRISPR-Cas system is especially useful in bacterial strains that have acquired some membrane proteins just for something, for pathogenesis, for stress resistance, for any other thing? So is a CRISPR-Cas system the way that the bacteria have to keep those membrane proteins? And why CRISPR-Cas? Because possibly these membrane proteins can be phage receptors, right? So that the picture it shows. Now, the membrane proteins are phage receptors and they are useful, and just to keep those membrane proteins, those phage receptors in the genome, we need to be able to fight against phage. Okay, so basically, I want to make a very long story short. We just checked that not all the strains have the same membrane proteins. We can always find blocks like clustered. So in this system, on this Baumannii system, so we have like two main clusters with different membrane proteins, and if we check here, we find different prophage in the genomes, and we find different spacers, right against those phage that we find in the genome. So things are starting to get clear. So we have, we have blocks of strains with this group of membrane proteins fighting or showing the same prophage in the genomes and the CRISPR-Cas spacers against those phages. And those are different from the other cluster. Okay, so here I present two examples of that. In Baumani again, the cluster 1 shows this protein, G, for instance, one of the membrane proteins, so in the strains that have these proteins but do not have a CRISPR-Cas system, so many of the strains with the protein carry also the prophage, while in the strains that carry the CRISPR-Cas system, then we don't really see a lot of the prophages. Same thing here in cluster 2. This is a protein thought to be involved in some kind of type 6 secretion system. I think we'll hear about those later today. And we have found the same pattern. So if we don't have the Cas system, then we can find phage there in the strains. If we have the phage, the Cas system, the presence of the phage is really rare. So these two clusters behave differently in the phylogeny. So the first cluster, the cluster 1, is widespread, doing this hide and seek game in the phylogeny. But the cluster 2 seems to be recently acquired, seems to be related to a branch of the phylogeny, and this has helped us to zoom in a little bit and to show this nice graphic in which we have expanded this phylogeny. So and we can find that in some point this group of strains, all related, right? In some point they gain this membrane protein. Okay, so in blue we have all the strains that carry these membrane proteins. In some point it seems to be to appear to show up the phage that maybe in our model, maybe using this protein as a receptor, and then we have this bunch of strains that carry the membrane protein and the prophage. But in some point they acquire the Cas system, right? So the phage disappear, and then we have all these strains with in red the CRISPR-Cas system, and in gold the spacers of the repetitions, of the repeats, against the phage, right? So in this point, we think we're pretty much confident that this is at least one of the biological significance of the CRISPR-Cas system in bacteria. So now I'm listening Pepe's voice whispering in my ear, and all of you will know what he would say now. Okay, Andrés, this is a nice story, this is a nice correlation, now go to the lab and show it, right? We are now starting a collaboration, so Ruben is here in the audience. I'm very happy that he's here. We're starting a collaboration, just trying to explore the possibility of this in the lab, right? Of this possibility of this model. So they are doing the mutants, and we have identified one of the prophage that belong to this cluster. So we have like 200 strains in which the prophage is basically the same 99% identity. We can find the ATT sites in the ends, and we are pretty convinced, confident that we will be able to induce the prophage and try to purify the phage and try to check whether this protein in fact acts like a receptor. Okay, so this is the story I wanted to tell you. That's the last thing I talked to Pepe. I would like to tell him in person. Now he's not here, so I'm telling this to you folks. This is the acknowledgement, the place where we are doing. Finally, we have this story. Well, this is part of a large story. This story just accepted in Science Advances. You can have a preprint an earlier preprint here, so you can, if you want, you can go for that. And well, thanks to Pepe and to all of you for listening to me.